Super Bowl, and I can remember. But if you're a Broncos fan, I guess you're happy this morning. So we're going to get into regulation today, and we have two things to do first. First is I want to clarify on the exam regrades for the midterm. I just sent an email around to everyone that sort of lays out what you should do. They're not due until Friday of this week. You can hand them in on Wednesday in class or at office hours if you want. And so it just says, you know, what I expect when you write a regrade request. So please follow that if you want to make a request. We're talking about we're still finishing up the pathways, right? So the way I organize this is pathways, covering all the pathways first, and then we talk about how to get regulated. And so where we're at is the pathways that we've sort of alluded to a little bit. We've sort of hit some of these enzymes already. But it's kind of good to review and make sure that we have all the pieces together in one place. And then what I'm talking about here is the storage of metabolic energy, of sugar, in the form of glycogen, and the fact that that's a storage particle for energy. So if glucose is a fuel source, then a whole bunch of glucose is bundled together in a glycogen particle. Of course, it's also a fuel source, but it's a stored fuel source. There's only one molecule of glycogen, and, instead of, shall we say, thousands of molecules of glucose. And for osmotic pressure reasons, which you probably talked about last term, you couldn't store that much glucose as individual molecules without popping the cell open, basically. So that's why it's one very good reason based on physical chemistry for why you should have glycogen. Not to mention that it's kind of all in one place as a particle. So these are just showing you some glycogen storage granules that contain aggregates and include in those aggregates the enzymes that will break the glycogen down. So glycogen is our energy source for either aerobic or anaerobic metabolism. I should mention that if you have a E. coli growing anaerobically or some bug, some microbe, they're always single-cell organisms that are anaerobic, right? Or exclusively anaerobic, or able to do all their metabolism anaerobically. You can do your sugar metabolism that way, including building up the glycogen and breaking it down. Fats, on the other hand, there's no fermentation pathway for fats, so fats only work in aerobic metabolism. So even though the energy storage is smaller than for fats, just because fats are more reduced, so you can get more ATP out of a fat molecule, out of a lipid than out of sugar, it still has that limitation. So glycogen can be stored in the liver for dispersal through the bloodstream, to broken down the glucose and then sent out. We just sort of alluded to that last time. There are glycogen particles in lots of different kinds of cells, though, not just in liver. Okay. So glycogen metabolism here, I've had this slide. This is really the sort of same slide you saw before in terms of feeding pathways. But here's glycogen here. And so this is really just the breakdown of glycogen in two different ways that we discussed before. But you will note from this slide that there's a gap between glycogen and the glucose pathway in the sense that G6P, we're going to go to glycogen and we're coming up from gluconeogenesis, right? We get to G6P. If we want to make glucose, for example, for export for a liver cell, then we would run the phosphatase reaction to go from here to here in gluconeogenesis, which would be the reverse of this hexokinase that's depicted. But if we're a liver cell, if we're a liver cell that has already sent out as much glucose as it needs to, or else we're a muscle cell that wants to store it, or some other cell that wants to store it, then we would go G6P to G1P by the reverse of this mutase reaction, and then we would build up glycogen with other enzymes that I'll show you in the next few slides. Okay. So the breakdown of glycogen, so we talked about the breakdown and the buildup. So the breakdown is phosphorylase. We talked about this enzyme already in terms of regulation, phosphorylation versus, it's a phosphorylated enzyme, and when it's active, it can be dephosphorylated as well. The cleavage is a phosphorylasis, so attack of inorganic phosphate. So one of the oxygens of inorganic phosphate would attack this C1 here. and it's coming from the non-reducing end. Right? This is the reducing end. This is the non-reducing end where there's no free OH group. So that breaks off glucose 1-phosphate. Now, glycogen is branched. So in addition to the enzymes that directly synthesize it and break it down, we also have enzymes that put in branches and remove branches. So if we're breaking down glycogen, here, the first step is phosphorylase that we just looked at. So that'll knock off individual glucose 1-phosphate monomers, here they are, coming off of the non-reducing ends. But because the molecule is branched, we also have these alpha-1,6 linkages here. And at some point, when you get too close to a linkage, the branched structure is weird for phosphorylase to try to handle in its binding site. It kind of wants some linear sugars, some number of them in a row, maybe 3 or 4 of them in a row. So if it gets too close to the branch, it kind of says, I can't go anymore, because it didn't bind well. And that's when another activity called the debranching enzyme comes in. And the debranching enzyme has actually 2 activities. One of them is to move every unit but one onto the non-reducing end to make that end bigger. And then the other one is to take this alpha-1,6 linkage and then just split it off into glucose. Okay, so when it does this, it's actually generating glucose directly. So not very much of the glycogen is broken down to glucose directly, because obviously most of it is going to be over here in G1P, but a very small amount of it is because of this debranching activity. Right? So now you've got this unbranched chain, what's left over here can then be chopped up more by phosphorylase, making more G1Ps, right, until you get to the next branch, etc. And then finally, we need mutase. Because if we're breaking down glycogen in a cell, we want to presumably run through glycolysis. Right? So we have G1P phosphoglucomutase, that's the enzyme with the equilibrium constant of 19 that we've been using for thermodynamics examples. Actually, we'll mention it again later today for that reason. But phosphoglucomutase is an enzyme that has a phosphorylated serine on it. So a pre-existing phosphorylated serine is in the active site of the enzyme, and there's a 2-step reaction. One of them is to take G1P and to make 1,6-bisphosphate compound by doing a phosphoryl transfer from the enzyme to the 6th position of G1P to make G16BP. Right? This is just an intermediate. It's on the enzyme. It's not diffusible. It doesn't go away. The next step is to transfer the phosphate at the 1 position back to the serine. Okay? So we've seen something like that already when we looked at the mutase enzyme in step 8 of glycolysis. Okay? So just a little bit more about that. Now, if you're a liver cell and you're breaking down your glycogen to glucose, the enzymes that we talked about are cytosolic. And so you'll get glucose, and you can convert it to G6P, right, using the mutase. But the problem would be then that if you're in the cytosol and you've got G6P, you know, what's going to prevent it from going through glycolysis? But if you're a liver cell, because you live for others and you're very generous, you want to give your glucose away, and you want to send it out to the bloodstream so that the muscles can have it. So you don't want to be breaking down that G6P you just pulled off of the glycogen. So the mechanism to separate it out is that there's a transporter in the membrane of the endoplasmic reticulum in the cell, which is just a connected network of organelles that is mostly discussed in cell biology courses as a trafficking pathway, that proteins get made on ribosomes, then they get translocated into the endoplasmic reticulum with a translocase enzyme on there for proteins that don't stay in the cytosol but that move off to other compartments in the cell. They might get stuck in the ER membrane and move through membranes, or they might get into the ER lumen and get translated to other pockets or left there, depending on their targeting signals. But here's another role for the lumen, at least in liver cells. It has it's in its membrane, it's got this transporter. The G6P that's produced after breakdown can be transported into the lumen, where it is hydrolyzed by a phosphatase